This is Immunology Unit 4, Lecture 6. We'll be covering Epstein-Barr virus. Here are your objectives. Epstein-Barr virus. In the United States, as many as 95% of adults between the ages of 35 and 40 have been infected with EBV. We become susceptible as infants when maternal antibody protection disappears. When infection occurs during adolescence or young adulthood, it causes infectious mono 35 to 40% of the time. EBV is an enveloped herpes virus, and therefore it causes a lifelong latent infection. Its DNA genome is double-stranded, making the virus more stable and less likely to undergo mutations. It can cause infectious mono, Burkitt's lymphoma, and nasopharyngeal carcinomas. You can read about the history. Next, we'll cover the structure. The genome structure, it's double-stranded linear DNA molecules in the virion. The size ranges from 124 to 235. The virion structure includes the core, which contains the viral DNA, the capsid, which is a icosahedral protein coat that surrounds the core, and the tegument, an amorphous material distributed between the core and the envelope. According to the CDC, 95% of the world's population ages 35 to 40 have been infected. 30 to 50% of adolescents and young adults infected with EBV develop infectious mono. Moving on to the pathogenesis, the virus is found in human saliva during an active disease state or during a period of reactivation that is asymptomatic. Most infections are a result of direct contact with an infected person's saliva. Human beings are the primary reservoir of EBV because the virus remains latent in a person throughout their life. EBV infects epithelial cells and human B lymphocytes. Epithelial cells become infected during the active phase of the infection and is the site for viral replication. Human B lymphocytes are the site of the latent EBV. EBV enters the B lymphocyte by binding to CD21, a receptor for both EBV and the C3d protein of complement on the surface of the B cell membrane. Once these cells are infected, the virus enters a lytic cycle characterized by viral replication, lysis of host cells, and release of infectious virions until the active infection is resolved. When B cells are infected, they become polyclonally activated, then proliferate and secrete several antibodies, including EBV-specific, heterophile, and autoantibodies. Several antigens have been identified in EBV-infected cells, and these include EBNA, EA, and VCA. EBNA is the induced nuclear antigen, EA is induced early antigen, and VCA is viral capsid antigen, which includes IgM and IgG. Antibodies to these antigens are a useful diagnostic tool and include VCA IgM, VCA IgG, EBNA IgG, and early antigen IgG. Infectious mono is a disease of the reticuloendothelial system that is evidenced by heterophile antibodies that disappear once the infection is resolved. Heterophile positive infectious mono is caused by EBV, while heterophile negative infectious mono is induced by EBV and other agents such as cytomegalovirus, adenovirus, and toxoplasma. The incubation period is 4 to 7 weeks, and infectivity continues for several weeks to months. The onset of disease can be acute or insidious and is characterized by fever, sore throat, and lymphadenopathy. Other symptoms may include hepatosplenomegaly, lymphocytosis with relative lymphs, and enlarged cervical lymph nodes. The acute phase lasts about 2 weeks and is followed by a lengthy convalescence. The patient usually develops transient IgM heterophile antibodies, has an abnormal white cell picture, and abnormal liver function tests. Generally, infections resolve within 4 to 6 weeks, although chronic infections do arise and are characterized by lymphadenopathy, fever, headache, pharyngitis, and persistent fatigue. The WBC count increases, there is a decrease in polymorphonuclear granulocytic cells, and a marked increase in monocytes and reactive lymphs. Here we have the lab testing overview, and next we'll talk about heterophile antibodies. Heterophile antibodies are antibodies that are produced in response to one antigen and that react with a second, genetically unrelated antigen. Examples include sheep erythrocyte antibodies in infectious mono, non-immune antibodies to the ABO blood group, and non-treponemal antibodies in syphilis. These antibodies in response to EBV-specific cell surface antigens during infectious mono react with erythrocyte surface antigens from sheep, beef, ox, and horse erythrocytes. Other heterophile antibodies that react to erythrocytes include serum sickness antibodies and Forsman antibodies. Heterophile antibodies appear in the serum of 85 to 90% of adults and adolescents in the acute phase of infectious mono. These antibodies reach significant levels by the end of the first week and peak at 2 to 3 weeks. They react with sheep red blood cells and are of the IgM class and represent an early response. Next, we'll cover the Paul-Bunno presumptive test. This is a screening test used to detect heterophile antibodies, which are not necessarily specific for infectious mono. They first associated infectious mono with sheep red blood cell agglutination and developed a test for the detection of antibodies. It is a hemagglutination test and employs a series of two-fold serial dilutions of heat inactivated patient serum mixed with a 2% sheep red blood cell suspension, resulting in two-fold dilutions beginning with 1 to 7 dilution concentration. After a room temperature incubation, the tubes are read for agglutination. The final result is the reciprocal of the highest dilution of serum showing visible agglutination of red blood cells. A titer of 28 or less indicates the absence of the antibody and a normal reading, whereas a titer of 56 or greater indicates the presence of heterophile antibodies found in infectious mono, serum sickness, and several rheumatic diseases. Positive tests are then subject to the Davison differential test to further differentiate the 3 heterophile antibodies. So this test is specific for the differentiation of infectious mono, serum sickness, and Forsman. So you guys need to study and memorize these 2 charts here and be able to answer questions based on these. We also have a immunofluorescent assay. This is a indirect immunofluorescent test that uses a sandwich technique to detect 4 antibodies, VCA, IgM, and IgG, early antigen IgG, and EBNA IgG. Results are read microscopically for fluorescence, and the titer is the reciprocal of the highest dilution showing specific fluorescence. We also have enzyme immunoassays. These are sandwich assays in which antigen is fixed to a well in a microtiter plate. Patient serum is added to the well and incubated. The wells are washed and an enzyme conjugated anti-human IgG or IgM is added. The microtiter plates are read spectrophotometrically, and the amount of color detected is directly proportional to the amount of antibody present in the patient serum. Here's a chart that shows when each of the antibodies are detected. We also have a few different slide tests I'll let you guys read about. and the monospot. The monospot uses direct solid phase immunoassay technology for qualitative detection of infectious monoheterophile antibodies in serum, plasma, or whole blood. In the test procedure, 10 microliters of serum or plasma or whole blood is added to the sample well. If the infectious mono antibody is present, it will capture in the antigen band impregnated on the test membrane. The developer solution is added to the sample well. As the specimen and the developer move by capillary action to the antigen band, the solution mobilizes the dye conjugated to the anti-human IgM antibody, and visualization of the antigen band in the test region will occur only when the antibody dye conjugate binds to the specific infectious mono antibody. A positive test is indicated by one pink band in the test region and the control region. This indicates the presence of infectious mono-specific heterophile antibodies. A negative is indicated by one band in the control region and no band at the test region, and an invalid test is indicated by no pink band in the control region. We also have membrane-based immunoassays and ELISA. This chart's also important, and you guys will need to study these too and be able to answer questions about them on your test. So that's all for this lecture. Go ahead and take the quiz on Canvas, and I'll see you in the next one.